is for reprogramming. I think we have also seen some things. What are all the ways or advantages and disadvantages of different reprogramming protocols? We were also in a place where we were trying to do genetic mechanisms or trying to understand the principles of genetic mechanism. In that, one of it I discussed was on using transposons and piggyback transposons. We can get into it later. If you want further details, then you can have a look at the paper and download things. Okay. So in today's class, we are going to start a new chapter, which is going to be a part of, one second. Which is going to be a technical paper. Okay. Like we have already seen some aspects. Is my audio, is my internet fine?
It went off a bit now. Now it's fine, sir.
Yes, please, somebody.
Can you hear me, sir?
There's a lag, sir.
Yes, now I can hear you. Okay. Let me try. I'll just restart this. Just 1 second. Am I audible? I hope you can see my screen.
Yes, sir.
Okay, let's go. Let's see how far we get with today's unstable this one connection. Okay, so this chapter is about. It is the title is actually it's a research paper from the book, which is actually a paper consisting of three different papers. Okay, it's a fusion of it, which is released as a book chapter. So pluripotent stem cell epigenetics and epigenetics during development and cancer. What we will discuss are some of the experiments and some of the results. We will not discuss all the experiments. The what I am expecting from you is that you know the principles of the experiment, how to do or what they are doing. And second thing is your ability to interpret. What the results are given in the book are not necessarily important, but this is the learning the principles and ability to interpret are the skills that I am hoping that you will acquire from this chapter. So first things, we will have a brief discussion on the epigenetics that we have discussed so far. And we discussed about DNA methylation, we discussed about histone modifications and so on. In that we came across CPG islands. There are several of them upstream of several within the regulatory regions of several genes. And the methylation is on the cytosine, CPG. C stands for cytosine, guanine, and P is the phosphodiester bond between these two nucleotides. So there will be several, whether that the CPG islands are methylated or unmethylated will determine the consequences on the genes downstream of them, or it could be upstream. So there is methylation or demethylation in that region, CPG island, we are saying. And there is also these regulatory regions could be several types, silencers, insulators, or enhancers, and so on, to which different types of proteins will bind, which could be, we'll just say, silencers, enhancers, insulators are the types of regions that we know. And to each of them, you may have a repressor binding or an activator or other proteins like CTCF, and so on. So the consequence, what happens to the gene, whether is it expressed or repressed, will depend on multiple things: the state, methylation state or not, and the type of region, the regulatory region it is, and what type of protein it binds. These two are actually the same thing. The terminology would depend based on the outcomes of the gene expression. Okay, so we discussed the DNA methylation generally represses transcription or expression because the protein or the transcriptional machinery that we have is usually to recognize sequences that are unmethylated. If this region was methylated, they cannot bind. That is the key way how DNA methylation does influence the gene expression. Not only that, we also discussed about how DNA methylation patterns can change the histone modification patterns and also eventually chromatin structure. I hope you remember this picture that is familiar to you. And this is a gene. Say, for example, this is where the gene is present, or open reading frame, which is usually CPG poor, in the sense it's not so much in CPGs. And the regulatory regions, like in the promoter, and these regions are usually, they have a lot of CPG islands or large concentrations of CG. So if demethylation occurs, that is de novo methyltransferases, they can methylate this region, and because of which the DNA is it becomes, I mean, it's methylated, and therefore the repression would occur. And upon replication, we would get something that is methylated. If it is upon replication, because of semi-conservative replication, we would end up something like this. So we have other type of methyltransferases like maintenance methylases, the NMT1 and so on, which will then identify these regions, and they will methylate the other strand as well, like that. Okay. And what also happens is, this is actually a much more, there are 4 nucleosomes in this place. But because of methylation and suppression and eventually silencing, you will have very compact ways of how the chromatin will become. I think we remember, I hope you remember, there is a picture that we have seen before, right? And we also discussed about genomic imprinting. And in imprinting, we discussed about IGF2 and H19. IGF2 is expressed from the paternal allele, whereas H19 is expressed from the maternal allele. So the region, differentially methylated region, DMR, which can be bound by CTCF like this. So there are enhancers which can activate either IGF2 expression or that of H19. These enhancers, what do they do, or which gene are they allowing the expression to, will depend on the methylation status of this region. If there is no methylation, CTCF can bind to it, because of which the enhancers or the activators that bound to these enhancers cannot interact with IGF2. This obstructs the interaction. So the activators allow the expression of H19. And well, this is what happens from the maternal allele. So we have IGF2 repressed and H19 expressed. Similarly, if you see the other one in contrast, in the paternal allele, the DMR, differentially methylated region, I'm not calling it domain, DMR is methylated, because of which CTCF cannot bind to DMR. So the enhancers or the activators bound, they can activate IGF2, and IGF2 is expressed, and H19 is not expressed, because this region also gets methylated, because of which there is no transcription. Moreover, the activators are not interacting, so H19 is not activated or expressed. So if we see as a biparental one, we have maternal and paternal alleles. IGF2 is repressed, H19 is expressed. From the paternal allele, IGF2 is expressed and H19 is repressed. And so in total, we have 1x of IGF2 and 1x of H19. There is a balance in that. Sorry. So we also learned about the imprinting status. What happens? We learned a general understanding of when imprinting happens, what happens, and so on. So we should also remember that there will be deviations from it. Different labs working on different genes may have differences, and that is also one of the part of what we are going to discuss in this chapter. The first one is, to start with, we have primordial germ cells with imprints and inactive X chromosome, with, if it is a female, and erasure of it during somewhere when they start moving from upon formation of primordial germ cells, they have to move into the gonadal ridge, genital ridge. And when they are moving, then at that time, all the methylations, including the imprints, they are all demethylation would occur. and X chromosome reactivation would occur. There, upon reaching the genital ridge, they will multiply and colonize the gonadal ridge. And then upon gametogenesis, if it is oocyte or sperm, new imprints are established. That is, new sex-specific imprints. That is, if it is a female, it has a specific imprint to establish, and if it is a male or the sperm is forming, then it has a different imprints to occur. So what I want you to remember is at this stage somewhere, there are no imprints, right? And at this stage of completely differentiated gametogenesis, we should have had imprints established, at least in this sperm, right? In the egg, there is initiation, but it's probably still not yet completed. Why I'm not saying anything very clearly or very establishedly that this is what happens in this region or in this time scale is because there will be variations from one gene, one type of imprints that we study, or one gene. From gene to gene, model organisms and different labs working, we will have some differences, which we are also going to see in this chapter. Okay, I hope you remember this picture as well. Erasure, establishment of imprints, right? If it is the oocyte, and here in this sperm by the time. So in this chapter, the authors actually provide a lot of some experimental data. As I said before I started this lesson, we don't have to really know the exact answers, but what I want you is to know is the important thing is to learn the principles and to learn how to interpret it. Okay, that is my focus of this this particular chapter. So in this paper, the authors have, I think, taken 3 different papers and put together as one paper, which is to determine the timing of when does erasure occur and when is the imprint established. Okay, so basically we already discussed that erasure occurs at this time after primordial germ cell formation or stage or so, and establishment occurs when they are forming gametes. And the information as such has come from experiments that were carried out in using papers like these ones or experiments like these. And they also try to have molecular mechanisms. Some of them, there are 2 things that they try to provide us is with. One of it is DNA methylation, and the second one is methods to study chromatin structure. Okay. So we know about DNA methylation, local histone modification patterns, and eventually chromatin structure. This can influence the DNA methylation can influence histone modification, and histone modification can influence chromatin structure. So how to study them is the question now. If you are aware of restriction endonucleases from the previous chapter, I hope you remember that restriction endonucleases, they are endonucleases, which means they will cut the DNA in between like this. And usually they are sequence specific. First thing. Second thing is they are double-stranded DNA endonucleases. That means they will cut both the strands. The third point is, or I'm just making it, there can be different types of ends or overhangs. This is a restriction site for ECO-R1. ECO-R1 recognizes GAA, TTC. So I hope you can see GAA, TTC, the complementary strand. Always read from 5' to 3', 5' to 3'. And you can read from in this direction is the same as reading in this direction, right? GAA, TTC, GAA, TTC. That is a palindrome. So we can use restriction endonucleases to study DNA methylation status. We will get to that. So when EcoR1 cuts, it makes a cut here and it makes another cut here, because of which the DNA is broken like this. And we have these regions are called as overhangs. Okay. This is the 5' side, so you will call it as a 5' overhang. There are, I hope you will also realize that there is a whole subject on recombinant DNA technology and so on, which you would need to, which will have comprise working with genetic tools like this, cutting the DNA, joining DNA, and so many other things. Here we are only discussing what we need for the purpose, and still I think you will be able to understand good enough. So using restriction endonucleases, when I said there could be types of ends, different enzymes give different ends. They can give 5' overhangs. They could give 3' overhangs, or they could be blunt ends. Blunt ends is if, okay, if there was a cut like this, we would have ended up with having GAA. This would have been one DNA, and TTC and AAG would have been the other one. There are no overhangs, so you would call them as blunt ends. But please remember that EcoR1 always recognizes GAA, TTC, and it will always cut the same way as shown here. It will not cut any other ways. So when I say different types of overhangs, it is dependent and specific to the enzyme. Yes. So what can we do? Or there is another part to it. It is about southern blotting. You may have heard about it. Southern blotting, where we are going to, there are 3 or 4 steps. One is cutting the DNA. Second is electrophoresis. Third is blotting. And the fourth is probing, you can say. The objective of southern blotting is to find a specific sequence in a sample, in a DNA sample, actually. DNA sample. That means you have a large genome and you want to know if a particular sequence, nucleotide sequence, it could be a gene or it could be introgenic region or anything, if that sequence is present in the DNA or not. Okay, I will repeat it. The objective of southern blotting is to know if there is a certain sequence present within the given DNA sample or not. Usually when you isolate, you will isolate the total DNA, which could probably mostly include the genomic DNA, and assume that you want to find out if there is a particular sequence gene in that DNA or not. So as per with the figure that is shown here, you would have taken the genomic DNA and assume that it is present. It is having the gene of interest. All you need is, you didn't know that before, you have to prove that it is there. So what you do is you digest it with using a restriction enzyme, something like EcoR1 that we have seen in the previous slide. And then when you digest it, it will break everywhere there is an EcoR1 site, it will cut. And so the genome, the genomic DNA, which is large DNA, is now present in fragments. And these fragments can be electrophorosed, and you can have the DNA in one place. So in this picture, there is a mistake. Can anybody tell me what is that mistake? Anyone?
The smaller ones will be closer to the positive.
Okay, the smaller ones will be?
Closer to anode, say positive
Why?
side.
Why?
Because DNA is negatively charged and the smaller fragments move faster than the larger ones. So they move faster towards the positive end.
Why to the positive end?
Because DNA is negatively charged.
That is right. So this is the mistake. If they were to put this arrowhead here, and this is where the well was, this is where if you have loaded the sample, then the migration DNA is negatively charged, and it will move towards the positively charged electrode, isn't it? And but there is, of course, the other problem is that you should have had smaller ones. Smaller ones will travel faster, and the larger ones will travel slower, right? That is another thing. So this part of the um, picture is wrong, and I when I first saw it, I think there is a mistake, and I didn't change it because it will help us try to find mistake, and probably when we find mistake, we will remember it for longer time. Yes, good that you got all the answer correct. So in this case, you have to remember the charges. What is DNA? Negatively charged, and it would move towards the positively charged electrode. And what is the rate of mobility, or the speed with which it is moving? The smaller ones will move faster, and that is because of the concentration of the gel. If you have high concentration, that is proportional to the, I mean, the larger ones will find more resistance from the agarose gel, while the smaller ones will find it lesser, and they would just move easily. So the smaller ones will move faster than the larger ones. Okay. So the point anyway is that in this case is we are going to have, or maybe we'll just try to make a correction to this. So if we can make this positive and this as negative, okay, and the arrowhead can remain this way. Is that fine then? Is this better? Anybody?
Yes, sir. It is.
This is perfect, right?
Yes.
Arrowhead design, you have all you needed to change was the electrodes here. Okay. So what electrophoresis does for us is that it will segregate different sizes of DNA in the from increasing from highest to lowest. Okay, here you should assume that this is the well where your sample is being loaded. Okay, once that is done, we can do blotting. Blotting is nothing, but you can also, I'll try to send some videos on northern blotting. You put the gel here. I'm showing the cross section of it. On top of it, you put nitrocellulose membrane and then several paper towels. And you will allow the wick-like thing to take up buffer, and the buffer will move in this direction. When the buffer is moving, what happens is the DNA that is present in the gel will also move upwards. And as the DNA is moving, it will get stuck to the nitrocellulose membrane that we have put here. And then, so now the DNA that was present in the gel, agarose gel, is now stuck or blotted onto the nitrocellulose membrane. That is the blotting step. So first is cutting, next is electrophoresis, then blotting step. Now we need to find out which fragment has gene of interest. So we would have used a probe a probe is a nucleotide, polynucleotide, usually DNA, or it could also be RNA could also be used, but it is radio labeled typically. There are other ways of non-radio labeled methods, but there are usually randomly radio labeled. Okay, like this. And when you add it, it is going to bind to the region or the sequence wherever it is present and not to the other. And when you develop the radiography, autoradiography, then it will show up as a single band like this. That means if there is a band and in that one particular location, that means in that location, there is complementarity to this sequence of the probe that we have added. Complementarity is dependent on the sequence, isn't it? The hydrogen base pairing. That means the sequence that we are looking for is present in that the DNA sample there. That is the meaning of it. But we will also discuss, we will watch some videos and then we'll come back and discuss this again after you have had some more briefing on that. So this is how, if I took DNA and treated it with restriction endonuclease, it might typically look like a smear-like thing because DNA is usually very large. And sometimes we may have to use a pulse field gel electrophoresis. We don't have to bother about it right now. And after after blotting, what we will see on the after development of the autoradiography is we will see bands like this. So if there are two bands here like this, what does that mean? It means there are two of these, the whatever probe, this I've just picked up from a random experiment from somewhere. So it means that the probe is able to attach to DNA present in these locations. Why is it able to bind? That is because it is complementary. That means there are at least 2 copies, or at 2 locations in the genome, there are fragments that are complementary to the probe. That is the meaning of it. Okay. Don't worry if you find it difficult. As we keep going, you will get a better picture. And anyway, we are going to repeat. I'm going to repeat in the hope that, and also as the objectives say, I discussed, that I need you to know the principle and I need you to be in a position to interpret the results. Okay? So if you can take a paper and pen, it will be nice if you can do this along with me. Here is an allele. It's just a simple exercise. Here is allele 1 and allele 2 of a particular gene, say gene X. There is only one difference between them. That is, in allele 1 has a restriction site here, whereas allele 2 does not have a restriction site. So this is, say, for example, with EcoR1, EcoR1 site and EcoR1 site, whereas here there are only 2 EcoR1 sites. So if the DNA was treated with EcoR1, we will get a 3 kb fragment and a 2 kb fragment, whereas in allele 2, we will only get 1 5 kb fragment because there is no internal restriction site there. And the box rectangle-like thing that is shown here, that is the probe. Okay. Now, if we have a person is homozygous for allele 1, so that means this person had 2 copies, and in both you will get cut 3 and 2, and this is also the same. It will get cut in 3 and 2. So when electrophoresis is done, what do you expect? You will get 2 bands at 2 kb and another one at 3 kb, isn't it? These 3 will form, these 2 of 3 kbs will form 1 band. These 2 will form another band like that. But if there is a person with homozygous for allele 2, we would expect that this person is going to give on both the cases, because we're saying homozygous, the person will give a 5 kb fragment and only one band. The question here, why I'm adding a probe, right? Look at the probe here. It is covering the whole range for 3 kb as well as 2 kb. That is the reason why you are able to see both in after southern blotting. You're able to see both of them. Say, for example, in theory, I'm changing something else, and I hope you will get the point I'm trying to describe. I'm making a new probe. Okay, see if you can understand that. I'm making a new probe, and I'm giving you this. This is the probe I'm giving. How many bands will you see if the person is homozygous for allele 1? In that case, too, you will have 3 kb, 2 kb, 3 kb, and 2 kb. Cut will be made. When you see the bands, okay, you have done electrophoresis, all the DNA are here. But the probe is specific only for the 2 kb fragment, or it falls within the 2 kb region. It does not cover the 3 kb. Therefore, upon southern blotting and development of the film, you will find only a 2 kb fragment. What would have happened if it were allele 2? We would have gotten a 5 kb fragment, and this is where I'm showing the probe will bind. That means it would have formed only one band, which is of 5 kb. Okay? So it will be nice if you can draw representative diagrams for the above 3. What are the 3? One is homozygous for allele 1. One is heterozygous, allele 1 and allele 2. And the other one is homozygous for allele 2. So this is how the result would look like if you have drawn it. First one is, this is homozygous for allele 1. So you'll get 2 kb fragment and 3 kb fragment. We are considering this probe that is here. Okay. If the person is homozygous for allele 2, you'll only get one 5 kb fragment or band like that. If a person is heterozygous for allele 1 and allele 2, we will get 2 kb and 3 kb from one of the, that means this person has restriction site here, and on the other allele, this person has like this. So the two bands here from 2 kb and 3 kb are formed from this, the top allele, and the 5 kb fragment or band would have been visible from here. Okay, it is the same like what we have discussed with genotyping. There are simple, nice ways to look at them. Okay, do you have any questions here? If you have, please feel free to ask so that I can explain it again and better probably. If you're not going to ask, then I'm going to give you a premise for what I will discuss in the next class. Okay? This is going to get a little more complicated. In this case, we were discussing about DNA, whether it is methylated, whether it is cut or not. And now our goal actually is to study epigenetics. Epigenetics in the form of DNA methylation. Studying DNA methylation patterns with restriction enzymes. As I said, there are restriction enzymes, and you can have different types of restriction enzymes based on the methylation status of the DNA. So first thing, there are restriction endonucleases that are mostly sensitive to methylation. That means if it is EcoR1, it recognizes a sequence like this. Okay. If the DNA is methylated, say for example like this, then EcoR1 cannot cleave it. It is somehow inhibited from acting on this site by the methylases. By the methylation of this site. This is a restriction site for EcoR1, and it cannot cleave it. But if there is a sequence which is not methylated, then it can cleave it like this. Those we can call them as, or refer them to as, sensitive to methylation. There are others which are insensitive. That means irrespective of whether they are methylated or unmethylated, they would just cut the restriction site. They would just cut it. Okay, for those type of restriction endonucleases, it doesn't matter if the target site is methylated or not. There is also the third category, that is methylation dependent. That means they will cleave only if there is methylation. Otherwise, they cannot cleave. Okay, so just quickly get to it. Methylation, sensitive to methylation, that means will cleave only unmethylated DNA. Insensitive to methylation will cleave methylated as well as unmethylated DNA. When I say DNA, don't make anything specific, don't take it general. It doesn't cleave all DNA. All restriction endonucleases are sequence specific. That means they will cleave only that particular site. Like I said, EcoR1 can only recognize GAA TTC and cleave only at that in this way. If the sequence is not there in a DNA, then EcoR1 cannot cleave it. So before, I think I'll also want to tell you, and they're also hemimethylated. I hope you remember that as well. This is methylated. Upon replication, it may give rise to two DNA molecules like this, right? I mean, one strand methylated and one strand unmethylated. There are some more things, and that's why these whole techniques will be discussed at least twice so that you will understand better. I have one question. When you talk about restriction endonuclease, there is another way of classification, whether the target sequence, we said they are all sequence specific, right? What is their sequence? Like we are discussing about EcoR1, and we say it is recognizing 6 bases, 1, 2, 3, 4, 5, 6. 6 base cutter, we'll say it as. There are some others which are 4 base cutters. And there are others which are 8 base cutters. Right? This is not one, for example, is a 8 base cutter. EcoR1 is a 6 base cutter. DPN1 or South3A is a 4 base cutter. You don't need to memorize all these things. But I want you to remember the principle and your skills in interpreting the data. Okay? And last, before I want to also, one second. Okay, I will not take the technique per se, and I want you to also remember, yes. Say, we have to choose, we want to study methylation patterns, and we want to use restriction endonucleases. And DNA methylation happens majority of the time where CpG or GC concentration is very high. In that case, we should have chosen restriction endonucleases which have lot of CG or GC. If you look at NOT1, the whole recognition sequence is CG, GC, GG, CC, GC. You're finding a lot of CG in it. And that means it is likely to fall within the regions where DNA methylation or epigenetic methylation is happening. Okay, and that's why the choice of restriction used in this case, typically, not always, typically would involve a lot of restriction endonucleases which have CG, high concentration of CG. EcoR1, if you see GAA, TTC, we have G and we have C, but there are no CG or CC, right? So the probability of EcoR1 falling within the region of this DNA methylation is very low. So we will actually use several other restriction endonucleases that are useful for analysis of DNA methylation patterns. Okay, I'll stop here. If you have any questions, please let me know. And maybe I will post a video from last class, last year. You can watch it and come back, and then we can have a better discussion. Okay? Any questions here?
Sir, I have a doubt.
Yes, please.
While adding probe, do we provide the apparatus with required machinery for repair mechanism?
Repair mechanism.
If only if the probe binds to the strand well, we'll have a defined strand. So, do we have
No.
the proof checking?
No. We won't have this. This is completely in vitro, okay, and all of it will be. We will take DNA, okay, and we will cut it and run it gels, and then they will come like this. Assume like that. These are double-stranded DNA. And there are other steps of denaturation where the DNA strands are made to separate like this. I'm showing it like this just for sake of understanding. Okay, there are many things that fixing and so many other things. And then we are going to add a probe which has a sequence that is complementary to the gene of interest. And when we add to this, if there is base pairing, then it will base pair like this. And when we will wash off all the extra probe. So only way probe is actually there is if it is hydrogen bonded to the target sequence or sequence of interest. And when we develop the radioaudiogram or X-ray film, we will find the band is given by the presence of the probe. And the probe is present there because the DNA is the sequence, the specific sequence we are looking at is at that position. Okay. There is, can you tell me if there is any other question you had in this?
Yes, sir. Let's take this 3 kb and 2 kb example.
Okay.
So, if we break between the 3 kb strand and the 2 kb strand, and
Yes.
the probe is only a bit specific to the
Partial.
3 kb. Yeah. So,
Yes.
how does
You're
it work?
right. Absolutely. That's correct. So usually the probe is very long. It can be very small, and it can also be very big. So in such experiments, we are making about, say, I have taken a 200 base pair probe, 200 bases probe. So in 3, you have this, and in 2 kb fragment like this, and denaturation, everything occurred, assume that. And then we have a probe to, 1 second, I'll finish it faster. So the probe can still bind here because it's able to make 100 base pairs of interaction here. We are adding multiple probes, right? One type of probe, but several molecules of it. So in the other case, it might base pair like this. So this will give a band, and the 2 kb also will give a band. Is that understood?
But there are chances that this portion might be somewhere else in the given strand too, right? Because it
Which one?
necessarily needn't be the same spot that we desire to get.
Yes. I mean, if it is there, if you get 3 bands, that means there are 3 different locations in which has the sequence specific to that. Okay.
Okay, Zakar.
Um, so what you need to understand is the number of bases that are required to form base pairing here. Okay. If there are only 2 bases that are forming, 2 means at the most you may have 6 hydrogen bonds. Then the probe may not be bound to it for too long. There are several other steps that we haven't discussed in this. You need to have washing steps and so many others. And you also have to remember that people try out different probes first, and then they would have optimized that this is the size of the probe that is giving good, accurate information for the experiments and conditions I am using. And then I would use like that. Okay. As I was telling, if the probe is like I showed here, if the probe is the purple one that I've drawn is just there, then you wouldn't have this binding. You would only be binding to this fragment. Okay. So the probe here is overlapping the restriction site that is present here.
Okay, so the
Yes.
overhands need not be stabilized and stuff.
No
We
need.
can leave
No
it
need.
like
Yes, they can be like that. That's all right. Okay. Yes, I'll try. Please watch some YouTube videos on southern blotting, and then we will get to this again in the next class. It will be interesting because it's a way of trying to interpret data, and that is the skill that we all have to improve over the time. Yes. Thank you.
that. Sure, sure. Thank
Take
you.
care and have lots of fun. We'll see you in the next class. And think
Thank
about the woolly mammoth too. Okay?
Yes, sir.
Yes. Bye.
Thank
Thank
you,
you
sir.
so
See you.
Thank you.